Do you ever just want to make DNA? Well, that comes crashing down when you realize you have no idea how to. But what if I taught you the witchcraft of those who can? My name is Dr. PCR, and today you will become experts in polymerase chain reaction, also known as PCR. PCR is a scientific technique that makes copies of a DNA sample, allowing amplification of a very small DNA sample. Because significant amounts of DNA sample are necessary for molecular and genetic analyses, studies of isolated pieces of DNA are nearly impossible without PCR amplification. Its development was such a major development for humanity that the inventor was awarded the Nobel Prize in Chemistry in 1993. The process of PCR involves three main steps: denaturation, annealing, and extension. To understand PCR, we must first understand DNA, the substance containing the instructions that make us. It is a double-stranded molecule, with each strand consisting of a backbone containing repeating units of phosphate and a deoxyribose sugar. Coming off each sugar is a nitrogenous DNA base, which is like the code that programs for the proteins that make us. A single unit of these three compounds forms the basic unit of DNA, the nucleotide. There are 4 DNA bases: adenine, guanine, thymine, and cytosine. Each has a complementary base which it always binds to. Adenine is complementary to thymine, while guanine is complementary to cytosine. The strands bind to each other through hydrogen bonds between the bases, which are bonds that, in the case of DNA, form between hydrogen and nitrogen, and hydrogen and oxygen. During PCR, we require a DNA sample, 3 DNA nucleotides called dNTPs, Taq polymerase, DNA primers, and a PCR buffer solution in which the reaction takes place. To replicate the DNA, we first need access to each strand. Denaturation involves heating the DNA sample to a high temperature, around 95 degrees Celsius, in order to disrupt and break the hydrogen bonds keeping the strand together. This heating is conducted for 10 to 60 seconds, and the result is single separated strands of DNA which are complementary to each other. In annealing, DNA primers are mixed into the DNA. These DNA primers are short pieces of single-stranded DNA which are around 20 nucleotides in length. They are specifically designed with base sequences that bind to opposite strands of the template DNA at the edges of the region to be copied, and they provide a starting point for DNA synthesis. 2 different primer sequences are used in PCR, one for each of the complementary strands. The temperature is decreased to 45 to 60 degrees Celsius, allowing the DNA primers to form hydrogen bonds with a single strand of DNA. This lasts 30 to 60 seconds. Finally, extension occurs. Taq polymerase, an enzyme that makes new strands of DNA, is added. It is named after and sourced from the heat-tolerant bacterium Thermus aquaticus, which lives in hydrothermal vents. Taq polymerase is thus suited for these conditions, being very heat stable. This makes it ideal for extension, as the temperature is raised to around 72 degrees Celsius, a temperature at which human DNA polymerase won't function. Taq polymerase moves along the DNA template strand, binding 3 nucleotides with complementary bases in order to extend the attached DNA primer by forming strong covalent bonds between nucleotides. This continues until the polymerase reaches the end of the template strand, and the result is a new DNA strand complementary to our original template strand. Extension is conducted for 20 to 60 seconds. After every cycle, the amount of DNA is doubled, and there is an exponential increase in the amount of DNA produced. Z times 2 to the power of n copies of DNA can be produced, where Z is the number of copies at the start and n the number of cycles conducted. Now, you've probably heard of PCR's uses in the context of that forbidden P word, the COVID-19 pandemic. A PCR testing variant, RT-PCR, is a major testing method, as it amplifies single-stranded RNA, which the virus contains, rather than DNA. After they stick that disgusting swab in you, the sample is sent for PCR testing. If the virus is present in any quantity, then its genetic material will be copied and amplified until it can be detected. Congratulations, you've just received an isolation holiday in that case. But it's not just COVID. PCR testing is used for detecting many other pathogens such as HIV and tuberculosis, helping in disease control. It's also used in forensics. Any DNA left at the scene can be amplified and thus analyzed, helping in identifying victims and perpetrators. Finally, PCR is often crucial in genetics research. For example, DNA from fossils is often highly degraded and very little usable for research. Using PCR, scientists can take tiny amounts of DNA from ancient remains and make enough to map out the evolution of life. So you see, PCR is a whole lot more than just COVID-19 testing. Thank you for coming to my TED Talk.